This is part 3 of the chapter 1 lecture notes. And where we left off, we were going over the 3 domains. We have learned that there are 3 domains of living things. There's the domain bacteria, the domain archaea, and now we're on the domain eukarya. And within the domain eukarya, which what the organisms in this domain, what they have in common is all of them have cells that contain a nucleus that surrounds the genetic material, the DNA. So within the domain eukarya, we have 4 kingdoms. We have the kingdom Protista, which consists of protozoans and algae, slime molds and water molds, which are not classified as fungi. Even though they're called mold, they're not technically a fungus. So algae and protozoans and slime molds and water molds are the ones that you find in the kingdom Protista. This particular organism is a paramecium, which is a single-celled or unicellular protozoan. And one thing that you'll find is even though they're unicellular, some of these protozoans are all unicellular. But even though they're unicellular, they're much larger than a bacterium. A bacterium would be like a speck compared to a paramecium. So we have the kingdom Protista, which you're probably not as familiar with unless you've done a lot of studying of biology. Then there's the kingdom Fungi, which contains your mushrooms, the kingdom Plantae, and then the kingdom Animalia. For the notes on the domains and the kingdoms, we have domain archaea, the domain bacteria, and then the domain eukarya, which we divide into 4 kingdoms, the Protista, the fungi, the Plantae, and the Animalia. All right, there are also supergroups within the eukarya. I would be a little bit more concerned about you learning the supergroups when you get to Bio 112, if you take Bio 112. We cover the supergroups when we cover the chapter on the Protista, because the Protista are placed within certain supergroups. But I do want to point out one thing, and that is for plants are in the supergroup Archaeoplastida. So basically it would be, a plant would be classified as domain eukarya, supergroup Archaeoplastida, kingdom Plantae, and then the rest of the classifications, the phylum, class, order, family, genus, and species. Now, the fungi and the animals are listed within the supergroup Opisthokonta. Excuse me. So fungi would be classified as domain eukarya, supergroup Opisthokonta, and kingdom fungi. And then animals would be classified as domain eukarya, supergroup Opisthokonta, and kingdom Animalia. Okay, scientific names are universal and Latin-based. In other words, the scientific name is the same no matter what language that the scientist uses for their everyday language. That way, people who speak different languages will always use the same name for the organisms that they're discussing. So, the way that organisms are named is a process called binomial nomenclature, which means 2-part name. In a scientific name, the first word is the genus, and it's always capitalized. The second word is the species. It's always in lowercase. And then you put both words in italics, but if you're like handwriting this, you would underline both words. But if you're typing, you would put both words in italics. So the scientific name for human, so in other languages, the word for human is different, but the scientific name is the same no matter what language you speak, and it's a Latin-based name. The genus name is Homo, and that's capitalized, capital H. The species is Sapiens with a lowercase s. And so the whole scientific name would be the genus plus the species Homo sapiens, and that's all in italics. And then corn is Zea mays, and you can see how that's written here. Okay, so that's how you would write a scientific name, the genus plus the species name. Now we're going to go over the scientific method. We will be using the scientific method in lab, and you will need to understand the scientific method in order to evaluate certain experiments and that kind of thing. We need to make sure that we understand how a controlled experiment is conducted. For the scientific method, basically we use that to solve problems in science. There are some problems that we wouldn't use the scientific method to solve. Particularly, there would be questions that we have that would be hard to set up an experiment to study. Questions like, is there a God? Paranormal type questions. I know there's movies out, that have tried to study paranormal activity, but I'm not sure how scientific you can be. You can be to a certain extent, but the scientific method is better for studying things that can actually be observed and experiments conducted to study the question. So other questions that you wouldn't use the scientific method to study would be like, who is or which color is the prettiest, or which person is pretty, which person is ugly. These are opinion type things. So we use the scientific method to find answers about questions we have about phenomenon that we observe in our world, observations we make about the world around us. So biology is one of those areas where we use the scientific method to answer questions. And it always starts with an observation or observations, which you would have a question that would come from those observations, and you would make a hypothesis, which is a statement that can be tested. And that would be how you would design your experiment. You would design your experiment with a control and a variable, a control group and a variable group. And you would make a prediction on what you think the outcome would be of your experiment. You would collect your data, use statistical analysis to analyze your data. And then you would report your data, your results in a table or a graph of some sort, and you would make a conclusion, which is whether or not you feel like your data, your results support your hypothesis, or whether or not you want to reject your hypothesis. So here's a flow chart, excuse me, that I'm not even going to go through because it's more than I want to go into, but it just shows you different outcomes. But we will go over some of these definitions. So observations are when scientists use their senses to gather information. This would be, you can do measurements, you can just use your eyes to observe what you see, like whether or not an organism moves toward or away from food, that kind of thing. But you use your eyes, your ears, your sense of smell, and a lot of times observations are measurements, like I say. They can be measurements of time. You can count certain things, you can measure length or width or volume, all different types of things, pH levels. But observations are measurements and just things that you observe with your senses. And they would lead to, writing the observations down as data. The hypothesis is a tentative, and this is a definition you need to know, a tentative explanation for what was observed. And then an example is the discovery of the antibiotic penicillin. You develop your hypothesis through inductive reasoning, and you have to make sure it's a statement, so it has a period at the end, it's not a question, and that it is testable. All right. An experiment is the series of procedures that are designed to test your hypothesis. And to design your experiment, you use deductive reasoning after you've designed your experiment to make a prediction of the expected outcome. So another thing you need to know is the independent variable and the dependent variable in an experiment. So all right, so you need to know, so let's think of an experiment. All right, so let's go back to the antibiotic penicillin. All right, so penicillin was discovered because it was discovered that when they grew the fungus penicillium on a petri dish, that bacteria wouldn't grow on that petri dish around the penicillium fungus. So what we found out is that penicillium releases an antibiotic called penicillin. And that's where penicillin comes from. It's from a fungus called Penicillium. But what they would have had to do is have a petri dish with no fungus on it, and then a petri dish that had the fungus on it, and try to grow bacteria on both petri dishes. So the petri dish with no fungus on it would have been the control, and then the petri dish that has fungus on it would have been the experimental group. As far as the so what we would have been looking for is no growth on the petri dish that contained the penicillium. And that's what they found. They found that if the petri dish had that fungus on it, bacteria wouldn't grow on it. So in this experiment, the independent variable, which is the experimental variable, this is the one that you as the experimenter are controlling, that would be the presence or the absence of the fungus on your petri dish. So your independent variable would be, in this case, would be the presence of fungus or penicillium on your petri dish. The dependent variable is what you're measuring. And so the dependent variable here would be the growth of bacteria. You would measure how many colonies of bacteria that you could see growing on the petri dishes. And your prediction would then be, where is that? So after you design your experiment, your prediction is what you think is going to happen. So you are going to predict that there will be less bacterial growth on the petri dish that contains the penicillium, and more growth on the dish that doesn't contain the penicillium. Okay? So the factor being tested, the experimental variable, is the independent variable. And what we were testing is the presence or the absence of penicillium on the petri dish. And then the dependent variable, which I don't see on here anywhere, so I'll write it. The dependent variable is what you measure. The dependent variable in this case is going to be growth of bacteria. So in all experiments that we look at, that we read about, that we do in lab, we're going to probably go over which variable is the independent and which variable is the dependent. So once again, independent is the variable that you are testing, and the dependent is the variable you are measuring. Okay, so a test group is the group exposed to the experimental variable. We often call it the experimental group. And then the control group goes through all aspects of the experiment but is not exposed to the experimental variable. So the test group in our experiment would be the group that has penicillium on it. The control group would be the group that does not, the petri dish that does not contain penicillium. If a prediction happens, then our conclusion is that the hypothesis is supported. If the prediction doesn't happen, the hypothesis is rejected. And then make sure you know the difference between a scientific theory and a scientific principle or law. A theory is supported by a broad range of observations, experiments, and data, but a principle or a law really has no serious challenges to it. It's widely accepted, although theories are also widely accepted, but I would just say a principle or a law is stronger.